In this module, you will be able to learn about the inclusion complex synthesis of cyclodextrin with other molecules. You will be able to understand the inclusion complex synthesis, especially with the drug molecules and their applications in the pharmaceutical industry. You will be able to analyze the change in properties of the drug molecule as well as the cyclodextrin molecule after the formation of these inclusion complexes. You will learn about some methods to increase the efficiency of synthesis of these inclusion complexes. You will know about the safety of the complex that has been synthesized by cyclodextrins. Cyclodextrins, that is short form written as CDs sometime, they were discovered by a French scientist named A. Villiers. They are known as structurally related group of natural products formed during the process of bacterial digestion of cellulose. Cyclodextrins are cyclooligosaccharides which contain alpha-1,4 linked alpha-D-glucopyranose units, and they contain a sort of lipophilic central cavity and a hydrophilic surface on the other side. The cyclodextrins have a huge range of applications in varied areas of drug delivery and pharmaceutical industry because of their characteristic ability of complexation and other versatile characteristic too. The most commonly known pharmaceutical application of cyclodextrins is their ability to enhance the stability, solubility, bioavailability and drug molecule safety. In the previous module, we have discussed at length about the structural details of cyclodextrin and varied applications. In this module, we will discuss about the inclusion complex formation by these cyclodextrins. Cyclodextrins possess the ability to form inclusion complexes with varied drug molecules by taking up a drug molecule in its cavity. Some of the examples of marketed products containing cyclodextrins is given in the table. You can see the alpha cyclodextrin containing 6 glucopyranose unit is the smallest cyclodextrin which can encapsulate smaller number of drugs such as alprostadil or cefo-TM hexatil, whereas beta cyclodextrin has a bigger cavity containing 7 glucopyranose units, can encapsulate a larger number of drugs such as iodine, nicotine, nimesulide, nitroglycerine, piroxicam, etc. There are other functionalized cyclodextrin moieties which can be specifically encapsulating some other drug molecules. Shown here in the table are the derivatives of beta cyclodextrin such as 2-hydroxypropyl beta cyclodextrin, which can encapsulate drugs like indomethacin, mitromycin, cisapride, etc. Then there are randomly methylated beta cyclodextrin which can encapsulate chloramphenicol and sulfobutyl ether beta cyclodextrin which can encapsulate the specific drugs depending on its cavity size. We have also 2-hydroxypropyl gamma cyclodextrin. This is a derivative of gamma cyclodextrin containing 8 glucopyranose units and hence have a bigger cavity. More often, these inclusion complexes are formed by interaction of some lipophilic moiety of the drug molecule with the central lipophilic cavity of cyclodextrins. No formation or breakage of covalent bonds takes place during this complex formation, and the drug molecules are in rapid equilibrium with the free molecules present in the solution. The factors favoring complex formation are release of enthalpy-rich water molecules from the cavity and other non-covalent interactions such as electrostatic interactions, van der Waals interactions, hydrophobic interactions, hydrogen bonding, release of conformational strain, and sometimes charge transfer interactions. The physicochemical properties of bound drugs to the cyclodextrin are different from those of the free molecules. Similarly, the physicochemical properties of free cyclodextrin molecules vary a lot from the bound cyclodextrin molecules. There are various methodologies available for bringing the additive changes in new inclusion complex formed between the cyclodextrin molecule and the drug molecule. Some changes that occur on complexation are change in solubility, change in chemical shifts in their NMR spectroscopy, changes in the UV visible absorbance values, changes in fluorescence, changes in drug retention, for example in liquid chromatography, changes in pKa values or potentiometric measurements, changes in chemical stability, and the effects on drug permeability through artificial membranes. Methods to monitor the physicochemical changes that are occurring after the formation of inclusion complexes are applied to study the mentioned changes in aqueous complexation media. These changes are often expressed in the form of measurements of conductivity changes, determinations of a freezing point depression, viscosity measurements, and calorimetric titrations, etc. For example, shown in this figure is one drug molecule which is forming an encapsulation complex with cyclodextrin, and thereby its solubility is changed in the given phase. The classification of complexes based on their substrate solubility has been done by Higuchi and Connors, as indicated by these phase solubility profiles. We obtain an A-type phase solubility profile when the solubility of the substrate, that is the drug, increases with increasing ligand, that is the cyclodextrin concentration shown on the right hand side of this figure. The AL-type phase diagram is obtained when the complex is formed of the first order with respect to the ligand and first or higher order with respect to the substrate. And AP-type phase solubility profiles are obtained if the complex is first order with respect to the substrate, but the second or higher order with respect to the ligand. It can be difficult to predict AN-type of phase solubility profiles. Complexes with meager solubility in aqueous complexation medium are indicated through B-type phase solubility profiles as shown in the following figure. There are many methods known to synthesize inclusion complexes of drug with cyclodextrin molecules, including the solution method, the co-precipitation method, neutralization method, the slurry method, the kneading method, and the grinding method. For preparing cyclodextrin inclusion complex in solution, a large amount of drug to an aqueous solution of cyclodextrin is generally added. In the table given here, some methods have been stated to enhance the complexation efficiency of these cyclodextrin molecules with the given drug molecules. Let us study how we can enhance these complexation. First of all, let us know what is drug ionization. The unionized drugs do usually form more stable complexes than their ionic counterparts. However, the ionization of a drug increases its apparent intrinsic solubility, resulting in enhanced complexation. So sometimes, although it is not forming a stable complex, however, due to the increase in solubility, it leads to enhanced complexation. Another method is salt formation. It is sometimes possible to enhance the apparent intrinsic solubility of a drug through its salt formation. because salts are more soluble in aqueous medium. Another method is complex in complex. It is sometimes possible to increase the apparent intrinsic solubility of a drug through formation of metal complexes. That means the drug is first complex with a metal, and then this complex is encapsulated into the cyclodextrin cavity, hence the name complex in complex. Yet another method is the acid-base ternary complexes. It has been shown that certain organic hydroxy acids such as citric acid and certain organic bases are able to enhance the complexation efficiency by formation of a ternary drug cyclodextrin acid or base complexes. Well, we have another method to increase the complexation efficiency, that is polymer complexes. Water-soluble polymers form ternary complex with drug and cyclodextrin complexes, thereby increasing their observed stability constant of the drug cyclodextrin complex. This observed increase in the value of constant increases the complexation efficiency. So you can see in all these methods we are concentrating more on solubilizing the drug into the solvent medium. The better the solubilization, the more stable complexation or more efficient complexation takes place. Another method is solubilization of cyclodextrin aggregates. The organic cations and anions are known to solubilize the uncharged drug cyclodextrin complexes that have limited aqueous solubility. This will enhance the complexation efficiency during the preparation of, for example, a solid drug cyclodextrin complex powder. Another method is combination of two or more of the above stated methods. Frequently, the complexation efficiency can be enhanced even further by combining two or more of the above mentioned methods. For example, a drug ionization and the polymer method, or the solubilization of cyclodextrin aggregates by adding both polymers and the cations or anions to the aqueous complexation medium. So we can see that we have just understood various methods to enhance the complexation efficiency of drug cyclodextrin molecules. There is another type of encapsulation known as non-conventional cyclodextrin complexes. Cyclodextrins are able to form non-inclusion complexes and aggregates via self-assembly in addition to conventional host-guest molecular interactions. That means instead of one drug molecule getting into the cavity of a single cyclodextrin molecule, there is a possibility wherein many molecules of cyclodextrin can assemble together by non-covalent interactions and hold the drug molecule all together. This is known as non-conventional complexes. It is assumed that the mechanism whereby cyclodextrin exert their effects, especially their augmentation solubility, is via the formation of non-covalent dynamic inclusion complexes. There is a growing evidence that supports the important contribution of non-inclusion based aspects of drug solubilization by cyclodextrin, including surfactant-like effects and molecular aggregation. These non-conventional cyclodextrin complexes gain more importance in drug solubilization by means of molecular aggregation. A figure of one such non-conventional complex is shown here. Here each species are shown by a different color and shapes. For example, the green ellipse shows the free drug molecule, whereas military color ellipse shows a drug in a non-inclusion complexes, whereas the black ellipse shows drug in an inclusion complex. Empty bucket shows empty cyclodextrin molecule. A bucket with an ellipse, black ellipse inserted, is showing drug cyclodextrin inclusion complex. So this figure gives you an idea about the non-conventional cyclodextrin complexes. Now let us study about the drug stability, how these drugs are stabilized by formation of supramolecular assemblies with these cyclodextrin molecules. The feasibility of a pharmaceutical formulation can be limited by stability issues, especially for aqueous formulation of drugs that are prone to hydrolysis or oxidation. The reaction rates can be affected by inclusion of the chemically labile moiety of the drug into the cyclodextrin cavity. In cyclodextrin solutions, the observed degradation rate for a chemically unstable compound forming 1:1 complex will be the weighted average of the degradation rates of free drug and the drug in cyclodextrin complex. In first order and pseudo first order reactions such as hydrolysis or oxidation, the stabilizing effect will depend on three parameters like the cyclodextrin concentration, the stability of the complex, and the degradation rate constant for the drug degradation within the cyclodextrin cavity. Many studies have shown that stability of chemically liable drugs like steroid esters, alkylating anti-cancer agents, prostaglandins, prodrugs, and various other compounds can be significantly improved through formulation with cyclodextrin. The studies also revealed that cyclodextrin can increase the physical stability of various drugs. For example, the evaporation of volatile compounds can be significantly reduced through complex formation. The cyclodextrin will also reduce the denaturation of peptides and protein formulations. So now you have learned how the stability of drugs can be achieved by encapsulating them in the cyclodextrin moieties. Let us summarize what we have learned. We have learned that the cyclodextrins have a huge range of applications in varied areas of drug delivery and pharmaceutical industry, and it is because of their characteristic ability of complexation and other versatile characteristics. Cyclodextrins possess the ability to form inclusion complexes with varied drug molecules by taking up a drug molecule in their cavity, and we have various sizes of cyclodextrin that can achieve this goal. Depending on the size of the guest molecule, we can choose an appropriate cyclodextrin moiety, let us say alpha cyclodextrin, beta cyclodextrin, gamma cyclodextrin, or their derivatives to suit the case. The factors favoring complex formation are the release of enthalpy-rich water molecules from the cavity of cyclodextrins and other non-covalent interactions like electrostatic interactions, van der Waals interactions, hydrophobic interactions, hydrogen bonding, release of conformational strain, and charge transfer interactions. The methods to monitor these physicochemical changes, like the changes in conductance or the changes in pH occurring after the formation of inclusion complexes, are applied to study the mentioned changes of the aqueous complexation media. We have thoroughly discussed how to increase the efficiency of formation of complexes of cyclodextrins with these drug molecules. We have also studied the non-conventional cyclodextrin complexes, and they gain more importance in the aspects of drug solubilization by means of molecular aggregation. And we have learned that the feasibility of a pharmaceutical formulation can be limited by stability issues, especially for aqueous formulation of drugs that are prone to hydrolysis or oxidation. And there are many methods we have just discussed to improve the stability of these complexes.